Uh, Hello, everyone. Uh, I want to make sure if my voice is clear.
This is clear. Yes.
Okay, thank you. My name is Raheel Shahzad, and as you can see, at present I work as a postdoc fellow at Brain. Today, actually, I'm excited to discuss with you about the prospects of nanotechnology in agriculture sector. I support my presentation with a case study that we have conducted in rice using titanium dioxide and nanoparticles to actually boost the ultraviolet B radiations, the tolerance of the rice against the ultraviolet B radiations. So, the contents of this presentation actually will be um, simple. First, I would like to give a short self-introduction and then we can move on to the real topic. What are nanoparticles mm, and what are their classifications and why we choose the metal oxide nanoparticles are NPs. I will use the word NPs throughout the presentation to make it more easier to speak. So nanoparticles, whenever I say, I will say NPs. And then, as I said, um, I will support my presentation with a case study that we have recently. Actually, it's already published. So about the self-introduction, um, I have done my B.S. from Pakistan, from my hometown, but then I moved to China and I got my master's and PhD both from China, actually from the same campus, Huachong Agriculture University. It's a beautiful campus. Mm, I think in Asia, this university currently its ranking is 153, if I'm not wrong. So it's getting a good ranking. And if you see at a global scale, yeah, it's like between 700 top ranking university at a global level. So if specifically I talk about my PhD, I have completed my PhD from College of Life Sciences, from a very interesting department, because this department is like an integrated department of biochemistry and molecular biology. So we get the taste of both. A work experience, yeah, I also worked at Nanjing Agriculture University after completing my PhD. And Nanjing is another very beautiful city in China. And then I have some experience working in Indonesia as well. And as I said, now at present, I am at Brain. If you are interested, I would encourage you to go to my Google Scholar ID, which is registered under my name, Raheel Shahzad. And maybe you can have a more better idea what kind of projects I have done so far. So it will be better if you can visit if you are interested. My research interest, if we talk just generally, then I'm interested in using biotechnology and nanotechnology applications to improve the growth and yield especially in the context of this climate change scenario. But if we go into like specifics, then I'm more interested in utilization of metabolomics and transcriptomics analysis to deep to drive more deeper that what kind of pathways are involved in these sort of interactions. So that's a very, I think, short introduction about me. And then now I think we can move to our topic, which is quite interesting. Uh, that how these tiny particles with very dynamic properties, both physical and biochemical, can be utilized to boost the agriculture sector. And if I talk about more details, nanotechnology is very diverse. We can use nanotechnology in agriculture from different angles. Like, for example, people have been also working to preserve vegetables or the fresh fruits that we get by simply uh, making some coatings with nanoparticles. So that is another direction to use the nanotechnology. But as myself, many researchers are interested how we actually directly utilize the nanoparticles in the soil or in the plants using the foliar application, or even as a seed coating. Then what could be achieved through that? So yeah, there are many diverse applications. So because we have enough time, so I will go through some background, of course, uh, that nanoparticles actually were first discovered by a physicist by the name Richard Feynman, and he gave a very famous talk at an American Physical Society meeting, uh, and the speech title was "There is plenty of room at the bottom." So that was the first occasion where he uh, talked about the nanoparticles and their potential. Uh, and he also suggested or proposed that why we cannot write the entire 24 volumes of encyclopedia at the head of a pin. So at that much tiny level, uh, uh, the nanoparticles can be imagined. So if we talk about the nanoparticle size and dimensions, because this is the very basic feature of nanoparticles. Um, so if we see at the organic level compounds or molecules, that can be considered as a nano materials or nanoparticles. Could be DNA and protein level. Uh, so some people believe that or defined it that nanoparticles can be of size between 1 to 200 nanometers, while other believe that nanoparticles are between 1 to 100 nanometers. Uh, there are some examples of different dimensions of nanoparticles. I think we are going to talk about it in our next slide, that these different shapes and structures, why they are important, and they have different purposes. So if we classify the nanomaterial then it should be it should fall into between one dimension to third dimension, from zero dimension to third dimension. But if you specifically talk about the nanoparticles, then it fall into zero dimension zero dimensional nanomaterials, or 0D. Yeah, as I said, the size, there are different definitions of nanoparticles, or NPs. Uh, so some believe that it is between 1 to 100, and below 1 nanometer, people call them usually as atom clusters. If you see the structure, some have a solid morphology, uh, while others could be hollow or porous, and that could be serve a different function. Especially if we talk about the solid morphologies. Solid morphologies have a like more uh, uh, volume ratio, but size to volume ratio, but porous or hollow one have a more low size to volume ratio. And mostly the hollow one or the porous nanoparticles could be utilized for encapsulations. So these are some differences. If we talk about the strategies for nanoparticle synthesis, basically there are two: top down or bottom up. The simple idea is, in the top down, we use the bulk material, fragment them, make it nanoparticles. While if we talk about bottom up, yeah, we use the atoms and make them clusters to make nanoparticles, to synthesize them. So these are two strategies. Both of these strategies are actually affected by many factors. That could be hydrophobicity or hydrophilicity and many others. The material itself, the nanoparticles, from where the nanoparticles are made of, itself make a difference. A very basic difference between the top down and a bottom up is that in the top down synthesis, we can produce uniform nanoparticles with a well-defined size and shape. But if we use the bottom up, then it produces dispersed nanoparticles. So this is a very key feature. Another very important difference is that in the top down, you can use a different range of materials to synthesize the nanoparticles. But in the bottom up, you require special conditions and a special material to synthesize them. If we talk shortly about the methods for NP synthesis, yeah, we have physical methods and we have chemical synthesis methods. Green synthesis is actually derived from the same chemical synthesis, but yeah, we can categorize it as a separate category, because in this method we use the natural materials from plants or bacteria or alga or fungi. So green synthesis is eco-friendly and non-hazardous, and also it's more sustainable. So we have basically three methods for nanoparticle synthesis. And another point to mention is that green synthesis actually can change the surface chemistry of nanoparticles, because once we use the uh, biomolecules from the organic material, whether it's plant or bacteria, those molecules actually act as a capping agent to further stabilize the nanoparticles. Because the problem with the physical method is that it can easily make aggregates of the nanoparticles, which is not an uh, ideal case in using the nanoparticles, because if they become aggregate, and their efficiency decrease drastically. So we prefer kind of disperse nanoparticles. So that's why people prefer the green synthesis. Uh, there are many other factors that changes with utilizing the method of green synthesis. Uh, their the nanoparticle chemical reactivity and their binding sites also change. Uh, so, for example, if I give you example of chemical reactivity, because we have different functional groups from the biomolecules, like a hydroxyl or carboxyl or amino groups, and that actually can either enhance the reactivity of nanoparticles, uh, and then could facilitate their interaction with the target molecules. This is also important to mention what are the global trends in using nanoparticles research. Um, and this is a data from a published paper uh, recently in 2023, where they have shown that the curve of publications or research publications related to nanotechnology are increasing. If we talk about the economics, yeah, the market size is quite large, and it is expected to increase further in the next 10 years. But it include um, all the market segments. For example, if people are creating developing nanofertilizers or nanoherbicides, that is a one market segment. But if it when come to application and also including the end user, so all of it come to this um, budget range. Uh, there are many interesting nano-based products so far have been developed. Of course, depending on its application, uh, from for example, only few examples here, like agricultural liquids, and then we have also dry fertilizers uh, with the different formulation of macro and micronutrients. So depending on the purpose, people have developed different products. One very interesting patent, uh, uh, a very interesting nano-based product, which is actually used for coating the conventional fertilizer. So it's basically using as a coating. So nanoparticles as a coating, and you provide it to the conventional fertilizers, and they have checked that it's quite beneficial in enhancing the efficiency of the conventional fertilizer. So this is again a a patented product, and it's quite booming. Why is the application of nanoparticles gaining momentum in agriculture sector? Of course, because of the environmental pressure on agriculture. So there is a report. Uh, bear me if I have time. Okay, I have time, enough time. So yeah, there is a huge environmental pressure on agriculture. So on one hand, we have limited sources of common atoms, nutrient atoms that we use for making fertilizers. On the other hand, because we use it so extra quantities that can give us a huge amount of GHG emissions, the greenhouse gas emissions. So by 2050, there is a report that the GHG emissions will rise by 80%, which is a huge concern for many countries across the globe. This slide is also very interesting. And this is actually data taken from a paper which is published in ACS, Sustainable Chemistry and Engineering. So these authors have actually recalculated the efficiency of the conventional fertilizers. So we know that the urea and MAP are the most common conventional fertilizers for nitrogen and phosphorus. But what authors have done is that what is the final atom conversion efficiency for these common fertilizers? So that is very low, ranging from 5 to 8%. It is, if we talk in the simple language, then that means if you apply 100 kg of urea, then the plant is actually getting only 5% of a nitrogen. It's a complex calculation, of course. For details, you must, I think, read this paper. But if we like talk in a very simple language, then only 5% of a nitrogen is coming out from the urea fertilizer and utilized by the plant. So that's a very low conversion efficiency. So people have been working on that to directly or indirectly increasing the nitrogen use the nutrient use efficiency, which we call it NUE, the nutrient utilization efficiency. Some people believe that green revolution is ended because the curves are the yield curves are becoming flattening. I mean, whatever the input that have been used, the yield has come to a saturation point. So then people believe that there is a need of AgTech revolution, and nanotechnology is the part of the AgTech revolution. I think we should go through it quickly. We already talked about it, that we have finite resources. On the other hand, we know that the emissions are rapidly increasing. So there is a need to increase the efficiency, and that we can do by using the nano fertilizers. It's very interesting. This paper also recently published in Nature Nanotechnology. Using meta-analysis, these authors have actually checked that whether the nano fertilizers have increased efficacy and relative to the conventional fertilizers, and they found interesting results that somewhat between 18 to 30% of different nanofertilizer formulation have higher uh, efficiency than the conventional counterparts. They also have, this is another article published in Nature Food, where they have actually checked whether cost effectiveness of the nanofertilizers are higher than the conventional fertilizers. I will highly encourage you to go through these papers to have a look. If we shortly talk about it, then we can see that if for the same 8 tons per hectare yield, we can get it we can get it by only 40 kilograms of nanofertilizers. While for the same yield, the conventional fertilizers utilization is higher, which is 200 kg or more. So that's very interesting. Another point, just quickly, that the nanofertilizer application, how indirectly can increase the efficacy, and that is actually because of its effect on the microbial diversity. Let's go through this quickly, because I want to come to my case study. Okay, so we have utilized titanium dioxide nanoparticles, which is a metal oxide NP, and it has its own unique chemical and physical properties, high surface area to volume ratio, its quantum effects, whether optical or magnetic, or its mechanical properties, all are very unique to these nanoparticles. Basically, there are 3 methods to use nanoparticles in real life. That either we use it as a seed coating, or foliar application, or soil application. So we have used it as a soil drench method, and basically, whatever the procedure we use, the entrance of nanoparticles into the cell remains the same. That is 3 ways: either the electrostatic interaction, which can change the charge potential and they can enter into the cell, or their specific binding, or their direct penetration by membranes or by stomata. So if we come to the case study where we use the titanium dioxide nanoparticles, we were basically interested whether these nanoparticles can boost the UV tolerance. I think I will not go through this because we have a short time left. So UVB is another very concerning situation for future performance of the crops because of the depletion of ozone. So there are very few people who are actually working in this specific area, trying to boost the UVB tolerance, or try to understand the mechanisms to boost the UVB tolerance. So we have checked it, and it was quite effective. So we have done a very basic experimentation, and our focus was to check the transcriptomic changes. So at the level of genes, what difference they make. So our focus was that one. So yeah, we utilized the titanium NPs at the 100 mg per liter. And then we also have, on the other hand, we prepare the samples, treat them with the UVB, and then we also, foliar applications. I mixed the other research with this one. So foliar application of titanium NPs was carried out. And then we determined actually very basic parameters, morphology and photosynthetic related parameters. But our focus was the finding out the pathways, of course, through identifying the differentially expressed genes or DEGs. So yeah, if we talk about the very basic results, it definitely increased the biomass of rice seedlings as compared to the control plants, or as compared to the plants that receive only the stress treatment, the UV treatment. So yeah, and if we talk about the chlorophyll pigment contents, yeah, it was decreased in the, obviously in the UVB stress group, but then it was significantly higher in the group where which has received the stress treatment, and after that it has received the nanoparticle treatment. So we call it UV plus TN. We have also observed interesting results for some key parameters for photosynthesis. For example, in the efficiency of the PS2 reaction center, which is denoted by Fe by Fm, was decreased, which is have a very close association with the net photosynthetic rate. So yeah, we find that the UV plus TN treatment group has a higher efficiency of photosynthesis in terms of FV by FM. So this is some basic results. I encourage you to read the paper, because I would not be able to explain every result in details in this presentation. So yeah, we have checked the effect of the nanoparticles and photosynthetic enzymes, Rubisco and FB Pace, and some other osmolytes like soluble sugar and soluble proteins, and we find very significant differences. Again, we have also checked whether ROS generation is higher in which group. So we find obvious reduction in the UV plus TN group. Then, yeah, our focus was to check the transcriptomic through the transcriptomic sequencing, because this gives a lot of information about the pathways, the molecular pathways or biological pathways that could be how to say, that could be different among different groups. So yeah, we performed the RNA sequencing on the samples. We basically have constructed 12 RNA libraries, and the mapping efficiency was higher than 90%, which is which assures the quality of the data. Again, there are some other quality verification parameters that show that the sample to sample correlation is higher that also show a good quality data obtained from transcriptomic sequencing. And then the interesting part is the identification of DEGs, which is differentially expressed genes. So we find basically that the higher gene dynamics were observed in the group of UV versus control. And then we also have an average DEGs in the group of UV plus TN compared to control group. But the interesting part is to go deeper into this to find the candidate genes, which we have done it at some level. That, for example, we have find 691 unique DEGs in the UV versus control, and in the upregulated genes, while 321 were common across all groups. But then the real part is the going into the biological and to finding out the enriched biological or cellular processes, which we did. I'm sorry, it need a very detailed discussion. So I will just go through it, that we find some obvious differential patterns of between the different groups. And then we also perform the KEGG pathway enrichment analysis, and we find some interesting candidate genes. So if we are going to skip it. Yeah. So yeah, here, if we talk about the transcription factors, we find very dynamic changes between the three groups. If you see on my right-hand side, we have only taken the commonly expressed gene among three groups, and then we map them using heat map. And you can see a very different patterns, where in the UV compared to control group, if the genes or transcription factors are downregulated, on the other hand, in the UV plus TN, the same transcription factors are enriched, are upregulated. So these could be a very interesting candidate genes for our further studies. Total of 56 commonly expressed transcription factors were found among three groups, and then this is the heat map showing different patterns, although they expressed in all the three groups, but differently. And then we also find DEGs related to oxidative stress. Again, I'm not going into details because there's a lot of details in here. Yeah, but one thing is interesting, that we have also measured the glutathione enzymes, and the RNA sequencing results is quite confirming the results that we get for the enzyme measurements. Because here we find that the for example, the 10 DEGs belonging to the glutathione pathway, the 9 out of the 10 were downregulated in the UV group, while these DEGs, all of them actually were significantly upregulated in the UV plus TN group. So that is quite like confirming our results through the RNA-seq. And then we also have differences, our candidate genes in the cell wall metabolism related. I'm not going into details. There is a lot of details here. And then we also find differences in the hormonal transduction pathway, a total of 37 commonly annotated DGs were involved, but they show very different patterns. And so we actually can select some of the candidate genes from these patterns.
We have more 2 minutes, Raheel.
Okay, so there is also very important to validate the RNA-seq results, which we have done. So yeah, and through the qRT-PCR. So we have randomly selected 12 genes, and they also showed a similar pattern. So that is quite confirming the RNA-seq data. So if we sum up, then yeah, the titanium dioxide NPs have a significant role in controlling the expression of multiple genes, especially when we talk about the after exposure to UVB stress. And if we go through the summary, yeah, the transcription factors that were more obvious in all the other families were MEEP, BHLH, and WRKY. So these families genes were more prominent and have a very dynamic expression among the three groups. So we could have a very interesting candidates for further studies from these families. Similarly, for hormonal classes, we find that the IAA, ETH, and ABA candidate genes were differentially regulated, particularly in the UV plus TN. So that could be a very good candidates for the research. And finally, if we talk about the DGs in the glutathione metabolism, that I said that we also confirm it by measuring their pathway enzymes, and it was further confirmed in the RNA-seq, and we find very interesting candidates in this pathway that could have a very positive role in enhancing the UV tolerance in rice. Okay, lastly, that it is also important to mention that nanoparticle concentrations and plant species and the type of stress, all of these factors are important when you design your research. And finally, the field trials are crucial because most of these studies are actually under the controlled environment in a glass house condition. So yeah, that is another concern that must be keep in mind when designing your research. Anyway, this paper was published in the Journal of Plant Interactions. So if you are interested, you can have a look. But we haven't reached to the like final stage of using the nanotechnology because there are many concerns. There are environmental concerns, and there are regulatory hurdles, and the cost and scalability is also a very major concern, where which will define the adoption of nanotechnology for agriculture sector. And then, of course, the public perception also need to be how to say, need to be changed, or have to be more aware from the skepticism and resistance from adoption of these new agri-revolution technologies. So thank you very much. I know there was a lot of details, so I just give you a overall picture of what we have done so far. I welcome any questions.
Thank you. Yeah, thank you, Dr. Raheel. It's very interesting presentation. It's very comprehensive from the beginning, and then you also show us how useful this technology. So I'm sure that all the audience in here, some of the research group is also working on transcriptomic, and then a lot of things related with what you've done, and then I'm sure also they would like, I think, in the future working with you. I mean, collaboration with you. So I open the time now for the audience to ask to Dr. Raheel. I think we can also, people can put on the chat box or directly. Time is for the audience. Please, can you raise your hand and then you can directly also speak up, ask to Dr. Raheel. It's about 15 minutes because we already run out. Actually, we already used to another 2 minutes. So we only have 12 minutes. No, I think 17 minutes. 17 minutes in total. Okay. Please, anybody who want to ask? I think I have a question, Pa Raheel. If you open the chat in here, you can you open the chat
Okay,
box?
all right.
There is a question from Pa Nurfianto.
Okay,
So you could, okay, you can directly, I think, answer the question. Can you read that? Or I read it. What are the differences or advantages and disadvantages of synthesis and use of nanoparticle from metallic element and non-metallic element for agriculture? And the second one, can you pick up that question? And the second one, what factors influence the uptake of nanoelement by plants, whether applied through soil or foliar application? What the factor influence that? And then the third question, UVB stress can cause plant DNA damage. What is the methodology for analyzing plant DNA damage and impact of the application of nanoparticle? And then there's a fourth one. Many researchers are interested in application and in increasing plant secondary metabolite through stress treatment, one of which is UVB. Do you have any experience of this? Thank you. So there are 4 questions.
yeah. Thank you, Pa Nurfianto.
Yes, Nurfianto.
Yeah, actually, they are very interesting questions. That what are the differences or advantages and disadvantages of the synthesis and use of nanoparticles from metallic elements? Well, yeah, as you can see from the name, there are some differences, very obvious differences, like metallic elements have a higher magnetic, how to say, magnetic property, but non-metallic have a non-magnetic property. So that that is a that could be a very long debate, because if in the context of medical uses of the nanoparticles, then metallic and non-metallic makes a quite big difference. But for agriculture, I'm not quite sure what could be disadvantages or advantages in the agriculture sector. But yeah, if you uh, uh, go to the second question, what factors influence the uptake of nanoelements by plants? There are many factors. For example, through the foliar application, actually, there is no involvement of soil, direct involvement of soil. So in one of my slide, I also mentioned that the nanofertilizers, nanoparticles can actually interact very actively with microbes. So when you apply through the foliar, it is not very effective in those terms to activate that microbes. But when you apply through the soil, it can have a very obvious interaction with the microbes, and that can make a big difference. And about the DNA damage, yeah, there are many experiments that actually can be done to analyze the plant DNA damage. Uh, I don't want to go into details, but like methylation and this kind of experiments that you can do to really check, because if I'm not wrong, the methylation of the DNA only occurs when it is damaged. So you can check it by methylation of the DNA. Uh, so if the methylation is higher in the samples, yeah, you can check it by that.
The impact.
Okay. Is there any question left? How do you make
Or,
sure that the
beside the methodology to analyzing, he want to ask the impact of application nanoparticle. I think that's already obvious, yeah. You
yeah
already mentioned in your presentation. I think he can just read your paper. And then the fourth one is currently many researcher interested in secondary metabolite through stress treatment, one of which UVB. Do you have any experiment of this?
that is a very interesting question, because yeah, some researchers are actually uh, interested to increase the secondary metabolites through the stress treatment, especially the UVB. But there are many factors to consider. Secondary metabolites increase also to a threshold. Uh, so this is the first thing, that how much metabolites, secondary metabolites, should be there to provide that protection to the plants. Yeah. So, and also the UVB, in the case of UVB stress, because in the field, the UVB stress is like constant. While at the greenhouses, we uses a very severe stress treatment, but in a short time. So that should be differentiated when we talk about the secondary metabolites under the constant UVB stress or under the short term. So these kind of factors should be considered. And secondary metabolites, how much? That is another thing. How much is
Okay.
more effective?
Okay, there's another question. I hope later if Pa Novianto doesn't satisfied, can
Yeah.
ask again.
Yeah.
But there is a question from Pa Dilan Lowry. How did you make sure that the NP solution can cover the leaf evenly with a lower waste possible to minimize the possible influence on the transcriptomic data?
Yeah, this is
Evenly.
something very technical. Yeah. In the real field, what maximum we can do is in the glass house experimentation, uh, that we can just select the specific amount, and then we can select the nozzle that we are sure that it will, when we spray it, it will deliver that much amount. That's the only possible things that uh, can be used for uniform application as far as the foliar application is concerned. Because when you come to the soil application, obviously you provide a very specific amount and mix it with the soil. About the wastage, it's very hard question, because wastage, in any method there is a wastage. But what we are more interested is whether that amount could bring the transcriptomic changes. Yes, there are many studies that have shown that once you apply those concentrations, they can bring those changes. So that is the important thing. If the methodology that we use can bring the changes, that confirms the methodology. Thank you very much. If you have a question,
Yes.
if you are not satisfied, yeah, you can have private contact with me. No problem. We can
Okay.
discuss further.
Okay, thank you, Dr. Raheel, for your presentation. I think unnecessary for me to make a conclusion. Yeah, what you did already you mentioned, and I think everybody who quite interested with the subject could contact Dr. Raheel because he will be around for quite long time. I think in this center research. So I think thank you so much. So I will applause for Dr. Raheel. Thank you very much. So Dr. Raheel, thank you for your time. So unfortunately this time, oh no,